Hello, everyone, and welcome to this presentation on the evidence of evolution. I'm Dr. Wesley Swenson. You can call me Wes from Central Arizona College. Today, we'll explore the key lines of evidence supporting evolutionary theory. As we navigate these lectures, I will frequently ask questions. These pauses are an excellent time for you to engage with the content. I encourage you to attempt answering each of them. The benefit being it will improve learning through active engagement and help you gauge how well you've digested the content so you know what you need to study. Maybe you can even leave a comment below to see how you are tracking or to stimulate some ideas within others. So let's dive right in. First, what do you suppose we are looking at here? You may have thought this looks like a magnified image of bird feathers. While you'd mostly be correct, this is actually a fascinating fossil of a dinosaur tail, preserved amber from Myanmar, complete with feathers. These aren't modern bird feathers, they're from a non avian dinosaur, providing a direct link between dinosaurs and birds. We'll talk more about what that link is. Zooming out here, we can now view the tail structure. How does this relate to the topic of this presentation? Well, hopefully you said this is evidence of evolution. This discovery highlights how feathers, which we associate with birds in our modern times, actually originated in dinosaurs before birds flew the earth. Fossils are an important source of evidence to help us trace evolutionary changes over time, but they aren't the only source. Consider these dogs, for instance. They share a common ancestry with gray wolves from over 100,000 years ago. Imagine an ancient wolf population like the one shown here over generations. Variations led to the diverse breeds we see today. How is it that we have so much variance within the same species? The diversity in dogs arises from selective breeding, where humans choose traits to emphasize. But underlying this is a natural process, that is, genetic changes in populations over time. And how much variance can occur within one species? Let's examine some historical examples. The photos displayed along the top are from a 1915 book called Dogs of All Nations by W. E. Mason. It shows breeds like the Bull Terrier, Whippet, and Basset Hound. Compare them to modern versions below, and you can see that entire breed characteristics have shifted due to accumulated genetic differences within a single century. This is not just normal variation within a particular population or within this breed. For example, do any bull terriers look like the picture at the top anymore? Do any basset hounds look like the picture at the top anymore? No. Despite being deeply associated with evolution for reasons that we'll go into later, Charles Darwin was not the first person to recognize evolution. The concept of evolution was first observed and posited by ancient Greek philosophers over 2,000 years ago. Charles Darwin called evolution descent with modification. Douglas Futoyma is a prominent American evolutionary biologist of Stony Brook University. He studied speciation and population biology of herbivorous insects and their specific host plants. He defined evolution as change in the properties of groups of organisms over the course of generations. Finally, here we have the Wikipedia definition as change in the heritable characteristics. So, those which are not gained throughout one's lifetime from experiences, but rather those that are gained from genetically, of biological populations over successive generations. For our purposes, evolution is the fact that populations of organisms change over time, becoming genetically different from their ancestors. You might have noticed that one of the key themes here is we're talking about populations instead of at the individual scale. We can observe evolution in real time, not just in fossils or picture books. For instance, bacteria have evolved pan-drug resistance, meaning they're resistant to all tested antibiotics. This graph showcases the percentage of bacterial gut samples found to have resistance to several antibiotics over the time period of the year 2000 to just beyond 2012. This shows rapid genetic shifts giving rise to new traits. In this case, antibiotic resistance in response to environmental pressures. We can observe microcosms of evolution in the human population. Ellis Van Creveld syndrome is characterized by dwarfism, polydactyl natal teeth, and congenital heart defects. The rate of incidence in the world at large is about 1 in 60,000 to 200,000 people. But the incidence in the Old Amish order of Amish in Lancaster, Pennsylvania is 1 in 200, and as many as 13% are carriers, meaning they have the genetic information but don't exhibit the trait. All cases can be traced back to one couple that was part of the founding population in 1744. Marriage and reproduction within this small group has led to the genetic composition of the population to be transformed. Now that we have an idea of what evolution is, what is the theory of evolution? Before we can define that, let's review what a theory is. From the National Academy of Sciences, a theory is a well-substantiated explanation of some aspect of the natural world that can incorporate facts, laws, inferences, and tested hypotheses. How are things supported through science? Well, science has its roots in philosophy. It's a derivative of evidentialism, which argues that belief must be justified, and justification is based on observable evidence. This is chiefly done through the scientific testing of hypotheses and scrutinized through a peer review process. Broadly encompassing means it must describe a large set of phenomena. For example, it might be an observable fact that if I were to drop my laptop, it would strike the floor. But the theory of gravity is one that describes how all objects with mass are under the influence of a phenomena called gravity. Evolution explains a massive body of evidence, including controlled experiments, observations, comparative studies, etc., that come from many disciplines of biology. The evidence for evolution comes from multiple fields. Paleontology examines fossils over geological time. Biogeography looks at species distribution around the globe. Anatomy compares physical traits. Embryology studies development. And bioinformatics analyzes DNA and proteins and their similarity or differences from one another. We'll cover each starting with paleontology. Paleontology is the branch of science that studies fossils of animals and plants. Most organisms never become fossils because their bodies decompose or are eaten before they can be trapped in a layer of sediment, and special circumstances must have occurred in order for an organism to become a fossil. Over time, sediment layers build up and compress, trapping organisms that died during that period. With time and pressure, bodies mineralize into fossils, preserving a record of life. Can you think of some different types of fossilized evidence of life that you have seen? Shown here are some of the different types of fossils, such as imprints, petrified remains, or amber preserved specimens like the dinosaur tail we saw earlier. Relative and absolute dating is another type of evidence regarding the fact that populations of organisms change over time, becoming genetically different from their ancestors. Relative dating determines the age of a fossil based on its relative order in the geologic or archaeological record. using methods such as stratigraphy or rock layers, superposition, younger rocks on top of older rocks, cross cutting relationships, in other words, how geological features intersect one another. Absolute dating relies on the presence of the ratios of atomic isotopes found in fossils or surrounding materials. There is a certain amount of radioactive isotopes of elements that occur naturally in the environment. Over time, these elements go through a process called decay, by which they are transformed into other elements. For example, the isotope carbon-14 decays into the isotope nitrogen-14. If you have a sample of carbon, it takes about 5,700 years for half of the carbon to turn into nitrogen. By measuring how much carbon-14 is left versus how much nitrogen-14 has been produced, you can get an idea of how long a material has existed. In this paleontology example, we combine the use of fossils and dating methods to see the progression of whales from their ancestors. Indohyus was a plant-eating mammal that resembled a very small deer, but were interesting because it had these heavy limbs and a thickened middle ear bone that looked a lot like a whale's ear bone. This is important because whales and dolphins have specialized ear bones that look unlike the ear bones of any other current living mammal. Ambiolocetus had the same thickened ear bones and had other features that would have made it well adapted to an aquatic environment, unlike Indohyus. For example, a pocket of fat in the lower jaw that transmits sound to the ear. However, it was not a powerful or efficient swimmer, and the structure of hind legs that would have not done well for supporting weight on land. It lived a lifestyle like a mammalian version of a crocodile. Remingtonocetus was missing an important ear structure that mammals use to balance on land, and had adaptations in the pelvis that would have allowed for efficient swimming, but as you can see from this image here, could not be effective for weight bearing on land. And finally, Basilosaurus and Dorodon had very tiny, non functional hind limbs that are characteristic of modern whales, flipper-shaped front limbs, and a branched tail like whales. Similarly, horse evolution shows toe reduction over time. Hyracotherium had four toes, as can be viewed here at the bottom of this graph. And Myohippus reduced the fifth digit, and its third digit became more prominent. Merychippus continued this trend of reduction of its second and fourth, while the third digit became leading, until the modern single-toed horses that we have today, with the second and fourth digit fusing with the third digit. And this provided more efficiency in travel throughout the grasslands and more support for their larger body weight. Transitional fossils fill gaps, like Archaeopteryx from 150 million years ago in the late Jurassic. Archaeopteryx was considered to be one of the earliest examples of primitive birds. It did have flight feathers, claws for perching, and tail feathers that indicated the ability for flight. However, it had less streamlined, short, rounded wings. It lacked the keel that modern birds use to attach their flight muscles to, and had less range of motion in its breastbone for flapping. It also had a long, bony tail, making the bird less specialized for flight than today's modern examples. So, again, transitional fossils exhibit traits that are common to an ancestral group and a descendant group, and they form these evolutionary linkages between the two. Another transitional form is Tiktaalik rosea from about 375 million years ago. This organism is an evolutionary bridge of fish to tetrapods. As you'll learn later this semester, tetrapods is a group that includes amphibians, reptiles, birds, and mammals. It had scales and gills, but also weight-bearing fins like wrist-like bones, a flattened head, and a mobile neck, traits of both groups. Evolution explains a massive body of evidence. And moving to the next line of evidence is biogeography, which examines how living and extinct species are distributed globally, complementing the fossil patterns we've seen. The supercontinent Pangaea existed 335 to 175 million years ago. Geological and paleomagnetic evidence shows its configuration, explaining why certain fossils are found where they are today. Geological evidence indicates the lines upon which the supercontinent broke up, and continents that were adjacent have similar geologies. For example, the eastern coast of South America and the western coast of Africa. And if you look at this image here, or this video, you can see South America and Africa being separated from each other. Biogeography demonstrates the theory of evolution in part by aligning with evolutionary principles of species having shared common ancestors, such as distinct species developing in isolated regions, and through continental drift, where species that were once a part of the same landmass became separated and while they look very similar to each other and have similar evolutionary histories. For example, Glossopteris seed fern. Although it looks very similar to a modern fern, we know that this plant was not a true fern because it produced seeds instead of spores. In fact, these plants were the first to produce true seeds, and Glossopteris seed ferns are all on southern continents. As another example, Cynognathus were early mammals in Africa and South America, and their distributions are consistent with connected landmasses before drift. Moreover, species that are evolutionarily related also cluster together geographically. Whale ancestors are found in Pakistan and India, 70% of marsupials in Australia, early hominids in Africa, and 25% of fruit flies are unique to Hawaii, suggesting divergence after isolation. Next on our list for evidence of evolution is anatomy, which assesses physical traits, and embryology, which examines the process of growth and development, especially in their embryonic stage. This compares traits across organisms and builds on biogeographical patterns. Homologous structures are similar body parts that are from a common ancestor. And these are anatomical structures like vertebrate limbs that have the same basic bones in humans, bats, and whales that are adapted differently. Another example of anatomy is that nearly all mammals have seven cervical vertebrae, including giraffes and mice, indicating shared ancestry despite these size differences. In plants, angiosperms share homologous flower parts. Looking here at the bottom, we can see the sepals. Above it are the petals, and then we have stamens, so on and so forth. And these are shared across the species of flowering plants such as baby's breath and marigolds. In contrast, analogous structures evolve independently for the same function via convergent evolution. Convergent evolution is the process of two separate evolutionary paths leading to the same trait, function, behavior, etc. For example, birds and insects both have wings and they're both for flight, but they're structurally different. And while all birds share a common lineage for the development of wings over time within their respective populations, insects don't share that common lineage for the evolution of their wings. So, therefore, this is not evidence of a common evolutionary ancestry between birds and insects as a homologous structure that we showed in the prior slides. Similarly, some Euphorbiaceae plants, pictured here on the right, have barrel shapes and spines like cacti, but they're unrelated families that converged for desert adaptations. Cacti are in a distinct family called Cactaceae, and their evolutionary path for spines and barrel shapes were separate to that of Euphorbiaceae. Vestigial structures are these unusual, unused remnants of homologous to functional ones elsewhere, like wings on flightless birds such as ostriches. And the world is full of vestigial structure examples. For example, blind cavefish have eye sockets without eyes, a vestige of sighted ancestors. Snakes retain pelvic girdles, and these are homologous to lizard hips, but they're not functional in snakes. And here again are vestigial structures in whales, which have hind limb bones, which are vestiges of their walking ancestors that we covered earlier in this presentation. Even in humans, vestigial structures include the tailbones, ear muscles, and the appendix, maybe. But these remnants don't have a clear modern function. So, as I said at the outset of this lecture, I was going to provide some questions that hopefully would engage you a little bit in your learning so that you can assess how you're doing and to make sure that you're actively learning. So, this is checkpoint one, which states mammalian and reptilian hearts exhibit parallel anatomy. These are examples of what? Take a moment to see if you can figure out what sort of a structure this is, or maybe you need to go back and review the material. Checkpoint two. Hoofed mammals have dew claws that serve no function. These are an example of a. Moving along, let's talk about embryology. One of these is a human embryo. Can you guess which one it is? Well, embryology reveals homology early on. Shared traits of human, mouse, bat, chicken, and alligator embryos include four limb buds, tails, segmentation, and gill arches. Yes, gill arches, which are the series of bony loops present in fish which support the gills. As we can see, embryonic development is a hotspot in time for identification of homology and vestigial structures. During this period of time, biology has been careful not to overtweak things because minute changes can lead to disastrous consequences in the creation of an individual. Vestigial structures often appear in embryos, such as human tails and gill arches, horses with five-toed limbs as an embryonic form that fuse into one later on in development, or whale hair that is present in the embryo but disappears later on. And this is related to a specific type of genes we call homeotic genes. As a refresher, genes are segments of DNA that encode for typically proteins, and those proteins, amongst other things, control the development of the body or cells and structures. And so these homeotic genes, specifically, they control what we would call the master plan of the body. Things like the presence and order, number of major body parts, so on and so forth. They're called highly conserved. And what that means is that they mutate very slowly. The DNA that encodes for those proteins, they don't mutate very quickly. And this is because changes are often lethal. These homeotic genes are shared across many different groups. And what typically happens is that they're just overridden later on in development. So let's look at checkpoint 3 here, which asks or states, explain why many organisms appear similar during embryonic development. Okay, so getting back to these homeotic genes, um, sometimes they don't always work at overriding the master plan, and the result is what we call atavism. Atavism is the reemergence of a trait from an ancestral organism. It comes when mutations in the override mechanism causes an organism to revert back to the original traits that were passed down from the ancestral organism. And again, I'll give you a few examples. Humans can be born with tails. Yes, that's right. This is extremely rare, but sometimes that embryonic tail doesn't go away. And that's what you see on the left. Whales and dolphins can be born with hind flippers instead of just having tiny, invisible hind limb bones hidden on the inside. And that's what you see on the right. Horses can even have extra toes that failed to fuse with a main hoof, such as the case seen on the left. Snakes can be born with little non functional legs, like what you see on the right. Truly bizarre, right? But what explains this weirdness is the fact that these genes for these master plans are all the same. They're all plays on the same basic construction that have just diverged into different forms over time. Okay, checkpoint 4. Occasionally, humans are born with supernumerary or more than two nipples. This is an example of. And checkpoint 5, what does this say about the evolutionary history of humans? We're now moving on to our last area of exploration for evidence of evolution, which is bioinformatics or molecular biology. This is also the newest, most recent field in terms of technological advancement, whereas the other four fields have been studied for hundreds of years. The ability to determine the sequences of an organism's DNA and proteins has only existed on the scale of decades. However, despite being the most recent, molecular biology is the most powerful and definitive tool for establishing relationships between organisms. Because whereas some of these other fields may provide us with evidence that is hard to interpret in certain cases, DNA evidence clearly demonstrates the degree of relatedness between species, just like it demonstrates the degree of relatedness between individuals. We all know that the more closely related two individuals are, the more DNA they share. For example, you share more DNA with your siblings than you do with your cousins because your closest ancestor with your sibling is your parents. But the closest ancestor that you share with your cousins is your grandparents. The farther back you have to look to find a common ancestor between you and any given individual, the less similar your DNA is. This principle is also true across species, not just individuals, but the entire tree of life. When we look at the DNA of various organisms, we can see that the percent similarity gets lower the farther away you get in the tree of life. This table here shows you the percentage of DNA that is identical to humans in each of these organisms. In mice, 88%, I'm sorry, let's start with chimpanzees. 96% of the DNA in chimpanzees is identical to that of humans. We're only 4% different. In mice, 88% of their DNA is identical to humans. Zebrafish, 73%. Fruit flies, 47% identical to humans. Even rice, which is a plant, has 25% of its DNA still identical to humans. Now, the fact that chimpanzees share 96% of their DNA with humans does not mean that chimpanzees are the ancestors of humans. No, it does not. This is one of the most common misconceptions about evolution. The idea of evolution does not claim that humans evolved from chimpanzees. That would be like saying that you evolved from your cousins. Humans and chimpanzees are very, very distant cousins, but as far as different species go, they are closely related. In fact, out of all species in the world, chimpanzees are our closest cousins according to the DNA evidence. The next closest are gorillas, the orangutans, then various types of monkeys. And as you get steps further away, you have to go back further in deep history to find another common ancestor. Now, as you know from our earlier discussion, that the genes in DNA provide the instructions for making proteins. So consequently, we see the same trend in proteins where the more closely related organisms have more similar looking proteins. Of course, to actually see this trend, you have to look at a protein that is found in a wide diversity of organisms. One example of these is the cytochrome C oxidase protein. This is a protein that is involved in cellular respiration. And you may remember from last semester that cellular respiration is the process that cells use to produce energy in the form of ATP. It's the series of chemical reactions that break down sugars and transform their energy into ATP. And cytochrome C oxidase is one of the proteins embedded in the inner membrane of the mitochondria through which hydrogen ions are pumped during the very last stage of the process. I've circled the protein in this illustration that is meant to represent cytochrome C oxidase. So, because all cells have to produce energy, virtually all eukaryotic cells have this protein in their mitochondria. Plants, animals, protists, fungi, they all have it. So that makes cytochrome C oxidase a good candidate to compare across organisms to study how they are related. And this table here shows you how similar the cytochrome C oxidase protein is in a variety of different organisms compared to humans. The protein is 105 amino acids long, and in chimpanzees, all 105 of those amino acids are identical to the one seen in humans. In rhesus monkeys, all but one of them are identical. In horses, 12 out of the 105 are different. In tuna fish, it's 22 out of 105. And in thalcress, which is a type of mustard green plant, 39 of the amino acids are different. So as you can see with proteins, there is a similar trend that we see in DNA, that closely related organisms have a greater degree of similarity in their proteins. And as you get farther away from the tree of life, the similarity becomes less and less. Now, as I said, molecular biology is the most recent area to start contributing to our understanding of evolution. And when this technology came on the scene, in some ways it really shook things up because until DNA data was available, scientists were forced to come to conclusions about how related two groups of animals are based on their physical characteristics. So, for example, whales and hoofed mammals were classified into two different groups called orders. Cetacea was the name of the group that contained whales, and Artiodactyla was the name of the group that contained hoofed mammals. And when you just look at their anatomy, that makes sense. They look nothing alike. However, when DNA evidence came along, the scientists were actually startled by the fact that examining the DNA of these organisms, the DNA told them that whales and hoofed mammals were actually more closely related than just their appearances might lead you to believe. And so, since the 1990s, the two groups have been merged into one singular group called Cetartiodactyla. Another example of a relationship that was confirmed by DNA evidence is the one between dinosaurs and birds. In 2006, researchers in Montana actually recovered some unfossilized T Rex bone, meaning it was not just the mineralized mold of a bone, but actual bone material that had managed to be preserved for over 65 million years. They had this partially fossilized T Rex leg bone that was so large it could fit in the helicopter that they were using to transport it. So they broke it in um, half to make it fit in the helicopter, and they were using to transport it. Um, and they got it to the lab, and they were able to take a sample of an extract of collagen proteins from the bone, and they compared the amino acid sequence of the collagen to other organisms. What they found out was that 5 out of the 6 proteins were identical to the collagen proteins in chickens, but not to the collagen proteins in amphibians like newts and salamanders. Of course, just looking at a salamander chicken and a picture of what we thought dinosaurs might look like, you would assume that the salamander is more closely related to the dinosaur based on its appearance. But the sequence of the collagen proteins told a different story. It's actually birds that are the closest living relatives of dinosaurs. One other evolution puzzle piece that molecular biology has revealed is the presence of pseudogenes in organisms' DNA. Pseudogenes are non-functional pieces of DNA that are homologous to functional genes found in other organisms. They are basically the vestigial structure of DNA. They're genes that are broken, but they're still there. As an example is the gene gulanolactone oxidase. Gulanolactone oxidase is an enzyme that is involved in the production of vitamin C. That's what's shown here in the slide, the molecular structure of vitamin C. The gene for this vitamin C producing enzyme is present in nearly all mammals. However, the version of the gene that is found in primates, including humans, has catastrophic mutations. The gene is still there, but the mutations in the gene make it non-functional. This is why humans need to get vitamin C as an essential part of our diets. We lack this enzyme, and if we had it, our bodies would be able to make vitamin C on their own. So, if the gene is broken, why is it still there? It wouldn't make any sense for a broken gene to be in our DNA unless you look at it in the broader context of evolution and understand how it was passed down from a mammalian ancestor where it did work properly, but got mutated somewhere along the way. And now we walk around with this broken relic of a gene instead. So we just covered a lot of ground in a short amount of time. I think it's probably best if we recap here with a summary of the evidence of evolution that we've talked about. Within the fossil record, patterns of traits show a progression of time. There's a logic and order to the fossil record where you can see gradual transitions happen across long periods of time. Some of the fossils that we look at are transitional fossils. That is, they have traits that are shared by ancestral groups of organisms as well as the descendant groups of organisms. And they show how different groups are linked and related to each other. The geographical distribution of very old fossils matches what you would have expected to see when all the continents were merged in the form of the supercontinent Pangaea. But if you look at more recent organisms that existed after Pangaea broke apart, you see that related organisms tend to be clustered together. If you look at organisms' anatomy, you find homologous structures across related groups of organisms, as well as vestigial structures that lack a function at all, and these are especially common during embryonic development. Related organisms tend to look extremely similar during embryonic development because they have very similar homeotic genes that control their master body plan. But cases of atavism can show the reemergence of ancestral genes that are still hidden in the DNA. And then finally, DNA and protein sequence data from molecular biology confirms the relationship of organisms that we see in all of these other fields of study. And what explains all of these findings put together is evolution, that populations of organisms change over time, becoming genetically different from their ancestors. All of this evidence makes sense once you understand that all life is related. Younger species are descendant from older species. Organisms accumulate change and diverge from each other over long periods of time. And the organisms that exist today are all cousins with varying degrees of relatedness depending on when they diverged. So, this wraps up our discussion of evidence and evolution. And we're just going to finalize this with a few more checkpoints to try and gauge your understanding of the material. Checkpoint 6. In 1974, researchers excavated a fossilized skeleton of a hominid, a human-like primate, in Ethiopia. The skeleton was nicknamed Lucy and given the species name Australopithecus afarensis. The fossil was 3 feet 7 inches tall and an estimated 64 pounds. She was bipedal, meaning she walked upright on 2 legs, and her humerus, which is the long bone in the upper arm part of the arm, was 84% as long as the femur, which is the bone in the upper part of the leg. In chimpanzees and other apes, the upper arm is about the same length as the upper leg. Their humerus is 97.8% as long as the femur. But in humans, the figure is 71.8%. Our legs are proportionally smaller than our arms. So Lucy had characteristics that made her intermediate between other apes and humans. This makes Lucy's fossils an example of a what fossil between apes and humans. Next, many animals have a gene called CMAH in their DNA. CMAH encodes an enzyme that is responsible for creating a carbohydrate identity marker that is present on the surface of cells. So remember, we would have talked about this in the last semester in BIO181. One of the features of cell membranes is that they have carbohydrates sticking off of the outer surface, which is what is highlighted in the yellow boxes here. And these carbohydrates can behave like the cell's ID badge and serve as identity markers for recognition by other cells. So CMAH is a gene that makes an enzyme that makes one of the carbohydrate ID markers. But in humans, the CMAH gene is broken. It's missing a 92 nucleotide section of DNA that is present in other great apes. With those 92 nucleotides missing, the enzyme made from the gene is non-functional, and therefore the identity marker is not made. And the absence of this identity marker leaves human cells susceptible to being infected with the most deadly version of the microorganism that causes malaria. In humans, the CMAH gene would be considered a what? All right, that wraps up this first lecture. Thank you all for being with me.